Hey, hey. Hey, Jean.
Hey, how's it going?
Good. How are you?
I'm having a bad day.
Oh, no, that's not good.
Yeah, none of my tech's working, so I had to go back to my backup stuff that I have.
Yeah.
I had a nice camera, and I was going to do EO. It's an EOS Canon system. It does really good video, and it won't work. And it worked when I was at home, but not when I'm on travel. So
Yeah.
always this kind of things. The other thing I'm having a lot of think about is whether or not I can present and still see my notes. So that's going to be a big issue. So I got to figure that out. So the lighting in here looks pretty decent, or I can dim it with the with putting a cardboard over the light. But is this pretty decent lighting wise? I am trying to stand up to.
Okay, give me 1 second. We're getting some feedback, so okay. 1 second.
Let me make sure I got the right video on mute thing on too. Yeah, I'm trying to use this one. Oh, sorry. This is the microphone that seems to work the best. Test. Is this better? I've got 3 different mics, so we can try different ones, but I am underneath a air compressor that goes off once in a while, so that might be an issue. Hey, Michael. Okay, so see if I can do it with what I'm trying to do is not lose my my situation here. So it says I'm not enabled as the screen share. Oops, got my chat up too.
Yep, that's totally my fault. Just turn it on.
So this is my slides. And is there a way to share it so when I go into presentation mode, it's not showing everything? Mm-hmm. From beginning. Now, is there a way to be able to minimize this so that I can actually see my script notes on my computer? Okay. There's got to be a way to do it. I just didn't know how to do it. Okay. Is there a way that someone else could project this? And I have my scripts. I mean, I could follow my script and kind of go through it. I don't know if Betsy could do it or if Dean could do it. I'm trying to look here to see if I can do this. Yep. Yeah. That's one reason I sent it to Betsy and to Nedra just a few minutes ago, because I wanted to make sure it was possible. Hey, Dean.
Hey, Gene. Good morning to you.
Good morning to you too.
Is your glass half full this morning?
They were full. It was a full class, and they were behind schedule. So, and of course, all my technology I was just explaining to Nedra has not been working. So,
shoot. All right. right, we'll get it straightened away.
And I'm actually I'm sitting right next to the BSL-4, so apparently I'm right under the air compressor that goes off like every 15 minutes. I'm hoping
That's just going to make it more interesting and realistic. It's authentic, Gene, I think. Well, I'm trying to figure out video-wise and how I can see you, the screen, and myself, and I'm not seeing that yet.
When I do that, Nedra, what do you see?
I see the small tiles of your presentation.
You're seeing exactly what I'm presenting.
All right, welcome everyone, and thank you for joining us today. I'm Dean Gray. I'm Director of MRI Global's National Security and Defense Programs. And it's my pleasure to welcome everyone to our webinar series, Confronting COVID-19. So this is the first of 6 presentations that we're going to do over the next several weeks, 1 hour each, that we have planned to share our expertise. We're going to answer some questions. We're going to have some open and candid discussion about these topics during this critical time. And hopefully you'll find this helpful and also informative. Now, during our webinar series, you'll get a chance to meet some of our scientists who are leading experts in their field across a range of related areas. So that's going to include biosafety, sample testing, product development, therapeutics and vaccines, and also mobile laboratories and containment systems. For each one of these webinars, we're going to have the presentation, then we're going to follow that with an open Q&A session, and then we're going to have some time in smaller discussion groups before we wrap them up just before the top of the hour. I've been at MRI Global 18 years, so I know all of our speakers very well, and so I'm a little bit biased, but trust me when I say that they're excellent at breaking down really challenging topics, presenting information in an objective manner, and overall, these are really incredible people who are doing great work. And we're excited for you to be able to get to know them better through this series. Now, a bit of background for those who may not be familiar. MRI Global was founded right here in Kansas City in 1944. And we're a science and engineering research institute, and we've earned a reputation for expertise across broad areas of national security and defense and global health, including infectious disease research. So that's why we're here. That's why we're here generally, and that's why we're here today. Our speaker is Dr. Jean Ollinger to kick off this webinar series. And Jean is a principal advisor at MRI Global who has expertise working with select agents up to biosafety level 4 pathogens, and has been directly involved leading our efforts to prevent and control infectious disease outbreaks such as Ebola and other coronaviruses like SARS and MERS all over the world. He's going to focus his presentation today on what we currently know and don't know about COVID-19. And we think you're going to find it very informative. So it's my pleasure to have you all here with us today and to introduce Jean Ollinger.
Sorry, I had a little issue there. Hi there, everybody. Welcome. Thank you, Dean. Welcome to our participants for the first in a series of COVID-19 seminar sessions. We hope you find these presentations useful in your daily lives and also in your operations if you work in institutions. And we will follow this part of the presentation with some questions and answers, but I would encourage you to go ahead and start putting your questions and answers into the comment box. That'll help us, first of all, help you remember and help us remember the questions as they come in, and then we'll be able to answer those at the end of this presentation. So we've learned a great deal about this virus and disease and what's going on. We're learning a lot more every day. We relearn some old lessons learned in the past. And even with 20 years of plus experience working with viruses and outbreaks and pandemics, I find it difficult to keep up with the information as fast as it's coming to us. We're bombarded with press releases, with media events. Of course, there's the political aspects of it as well, but it's hard to keep track of what's new and, of course, what's repeatable. This leads to situations where we're unconnected data sets, and we're also often getting conflicting reports. And so a lot of my colleagues ask me about this, and they're like, why do we see that? And I remind them that's part of the scientific process. We do this as part of how we do science. Even incorrect or negative data sometimes turns out to be interesting data that helps us move in the right direction. So in the scientific process, it's a slow process. It takes time, but over time, we'll find the truth and the facts, and we'll move forward with that. So the data is reconciled as time goes on. In this presentation, I'll review some of the basics and discuss some of the areas of emerging evidence that are coming into what we're understanding about this virus and how to combat this virus and infectious disease threat. history repeats itself, and that's unfortunate. It's got good news and some bad news. I'll start with the good news. First, the good news is that history does repeat itself, and we can apply the lessons learned from other viruses, other outbreaks, and other pandemics and how we address this virus. Nearly everything we've learned about this virus, and I always say learned in quotation marks, is based on our experiences with other viruses, other pathogens, and especially coronaviruses. The collective history of coronaviruses and then, of course, other viruses helps us understand how to proceed forward with scientific and public health knowledge. And so the things that we've experienced, the lockdowns, or that's what some people call it, or the staying home and flattening the curve, those are all lessons learned from previous outbreaks that happened in the early 1900s. So this is not new knowledge, it's just being applied to our current situation. And what's interesting about this and what's the good news is that the things that our mothers taught us, at least my mother taught me, was that covering your coughs, wearing a mask, actually that's something that was pretty important back in 1918, as you can see in this slide. It's nothing new. And of course, washing your hands and resting when you're sick are all very good positive processes to follow when you have an outbreak or a pandemic. So these are all good news because they're all things we have control over in our daily lives. The bad news is that history does repeat itself, and with high fidelity. And so we're more likely to suffer struggles that we've experienced in the past, which is people not wearing masks. Of course, the issues with not washing their hands properly and doing things that they shouldn't do. And of course, we've done a good job at flattening the curve in most parts of the country right now, but we're starting to see a resurgence. So if you look at the news in the last couple of days, there's been resurgence in Arizona, Florida, and also in Texas. We flattened the curve, but the curve didn't go back down to zero. So we're still in the first wave of this outbreak. And so we're learning some lessons about how to deal with that. I'm a little surprised as a scientist that works with these viruses. Coronaviruses typically and flu and other respiratory infections tend to go away in the summertime. But again, this is a virus that we've never seen before. And so we're learning new things as it goes along. Unfortunately, lesson in history is that it does repeat itself. So we're likely to see a second wave or a spike in the first wave in the fall. So viruses, respiratory infections do come back in the fall, including flu and other coronaviruses and other viruses like RSV, respiratory syncytial virus. So we are likely to see more infections as time goes on. But with new treatments that are coming on, the new vaccines that will be available in early 2021, and good hygienic practices, we can actually defeat this without too many deaths and morbidity issues. So, and the goal with that is to reach herd immunity. I know it's a term that's used kind of a lot by epidemiologists and science geeks, but herd immunity essentially means that more than 50 to 70% of the population has been exposed to the virus and actually has an immunity to the virus. That really reduces the amount of people that can be infected and the transmission rates that occur as time goes on. Unfortunately, we're still probably in the 10 to less than 10% of the population who's been exposed. Oops. So rest assured, MRI Global has been working for many years, working on pandemics and outbreak responses, primarily in the preparedness and response phase. This is a cyclic process that we follow that all folks that work in public health and outbreak and pandemic responses work in. We try to be in the prevention and mitigation and preparation areas. That's where we spend most of our time. And when we spend time in those areas, we actually prevent what we're occurring right now, which is response and recovery. We're transitioning now between the response phase, where you actually detect the virus and start looking at it in patients, and then we're moving towards recovery, as you'll see in the later slides. Recovery phase can take a long time, as you will see. I will warn you that we follow this process because we know that the viruses that are most likely to impact humans are coming from animals, and so we follow the One Health Triad, which is a process in which we look at animals and the environment, and of course humans, to make sure that we're not having new viruses emerge. And so this is an important part of what we do. It's very complex. It has a lot of dynamics to it that are difficult to capture at one point, but it does lead to a protection. When we have a response like we're having now, we have both the primary response of the disease itself, but we also have secondary and tertiary effects of the disease. And that's seen in our economy and our social structures, and we're seeing that right now. This happens in every outbreak or every pandemic that we've dealt with in the past. And these secondary and tertiary effects really make the primary effects much more difficult to deal with. And so again, history teaches us how to do this and how to deal with this. It's our goal to make sure that we limit these unforeseen impacts and also address these primary issues such as detection of the virus, virus surveillance, sequencing, things like that as we move forward. And that'll stop and slow infections as time goes on. On a personal level, in this slide, I'd like to really emphasize something, that while COVID-19 is really, really bad, it is not the virus that keeps me up at night. The question I also ask my students all the time is, what keeps you up at night? And those things, what scientifically, what in your work life? In my work life, it's viruses that may emerge that actually have the potential to do really bad harm to society and actually cause high morbidity. And I would like to say COVID-19 was one of the cases, but unfortunately, the World Health Organization, WHO, and other experts, we're all concerned about this disease called disease X. It's the virus or bacteria that we have not seen yet. It's most likely going to be a virus because viruses emerge from pathogens, from mammals that then infect humans. And what we've known is that there's about 200 known viruses that cause human disease. There's millions of viruses that we know that exist in mammals. So this is a big concern of ours. And the death rates we see with COVID-19 and SARS-CoV-1 and SARS-CoV-2 are clearly something we don't want happening, but they're nothing like what we'd see with this outbreak that we expect from disease X. We would expect to see children, young adults, and actually the elderly equally impacted by morbidity and mortality. Fortunately for us for COVID-19, it's a certain group of individuals that are most likely to have a negative outcome from this virus infection. So again, this is a lesson for us to learn. We've learned it before. For HIV, for SARS-1, SARS-2 now, Ebola, MERS, Zika, and even influenza multiple times, we've learned that we can actually have a high death rate and high morbidity around the world if we're not careful. So again, we've lost those messages, and now we're being told this virus is really giving us another opportunity to relearn those lessons and apply those in the future, so that if we do the prevention, mitigation, and preparedness steps, we'll be better prepared in the future. But of course, we have to focus on COVID-19 right now. So I show you that slide to show you this slide, which is really kind of getting back to the history of MRI Global and what we've been working on. This is not our first rodeo. Rest assured that we've been working on this diligently to prevent and respond to outbreaks and pandemics. This has been going on for more than 20 years with regards to MRI Global, but MRI Global's chemists, engineers, physical scientists, and biologists have been using this approach for more than 75 years and using applied science and technology to make the world healthier. And this is one of our goals and one of our divisions that we've been working in. And we started to share this knowledge around the globe. We continue to share this knowledge around the globe, teaching and learning from our partners at the local, national, international level, and really looking for these naturally emerging diseases and stopping them before they spread. We've been a key player in making sure that we're protected against weapons of mass destruction as well, which sometimes are biological agents, and also similar, naturally occurring disease and disasters. So disaster response is important for us too. I like to tell people that our goal is to detect and keep the world safe from pathogens, and ultimately one day actually putting ourselves out of business. Now, there's plenty of work for us scientists to do, so that's totally possible. But again, this is a business in which we're busy working at this point. So, and then, this slide here is really an example of what we've done in the past. So for the outbreak in West Africa, we basically in 2014 and 16 deployed our teams. One of the big issues was, again, just like COVID-19, was diagnostic testing. It could take days to get a response when someone showed up at the Ebola treatment units, which are shown in this picture here, these long tents that are multiple tents in a row. We deployed our newly developed laboratory to actually do the testing right next to the Ebola treatment units. So what took days now took a walk across the parking lot to the location where we could actually run the samples, and literally had answers by the afternoon if they were if the sample got to us in the morning, which was a huge game changer within the outbreak response. We also developed containerized systems to actually transport responders who got infected while they were there, and we're able to bring those back, and you'll learn more about those in our conversations as we move forward. So these are all important things about our process. And of course, training. We train all the time. It was training our national partners, but also performing the tests while we were there. More than 20,000 tests were done in just one country alone. So we actually did a lot of testing while we were in country. I will say that as I move away from that slide is that recovery takes a long time. So we've been part of the recovery process now for 6 years in West Africa. So recovery does not happen overnight. And so we should expect time to change for our current situation, COVID-19, to take time for us to actually go back to normal. We have this new normal that we're hearing a lot about in the press, and that's really what we're focused on now for the next couple of years. So what is a coronavirus? Coronaviruses are a large family of viruses that include Middle East respiratory syndrome, MERS, which emerged out of Saudi Arabia, the Middle East during the 2013-14 time period. And then SARS-1 was a virus that basically very similar to SARS-CoV-2 that came out of the Asian countries and basically spread around the world pretty quickly. And both these viruses, unfortunately, fortunate for us, were very difficult to spread. So, and they also required high comorbidities for lethal or hospitalization cases. But I will remind folks that there's more than 4 common cold viruses that cause the common cold every year that are coronaviruses. We see these, they're on diagnostic tests already. So they're on systems when you can go to the doctor's office or the hospital and get tested for. And in fact, it's very common that people have a cold caused by coronaviruses. Unfortunately, these colds that are caused by these viruses don't really offer any protection against SARS, MERS, or SARS-CoV-2. So it is a very specific response your body needs to respond to these viruses. I always like to remind people too that there's over 2,000 known coronaviruses, mostly in bats, but also in cats and dogs and in humans, as we just showed here, but also even in dolphins. There's actually cold viruses that spread that are coronaviruses. So it's a wide variety of animals, mammals that actually can carry these types of viruses. And we learn more about those every day. And then this virus is a novel coronavirus because we had not seen it before, and it's been identified in humans, and it's spread pretty quickly. And because we haven't been exposed to it before, we've had a lot more disease spread that occurred in the US and around the world globally, making it a pandemic. So this is what really makes this a different virus than we had in the past. I've been quite surprised at the speed of which we're actually able to learn more about this virus. We're applying old knowledge about the virus to the current virus and learning more every day. And the speed in which the sequences are provided, the vaccines are being developed, and the treatments that are being evaluated in our animal models and our vitro models is just rapid. It's unbelievable. In the history of working with infectious diseases, I've never seen this type of worldwide global attempt to really find a way to work around this virus. So it's really impressive to see what's going on with this new virus. With this knowledge, we'll increase the probability of finding drugs, finding vaccines, and of course, finding ways to thwart the virus. And this is really being learned on a daily basis as we move forward. Oops. So I get asked this question quite a bit. A lot of folks have a misunderstanding about the virus, and they say, oh, it doesn't really matter. This is just another virus. And it is not just another virus. And this slide here really points to the reason. Respiratory diseases leading to pneumonia is still one of the leading causes. It's the #1 leading infectious disease cause of death in people around the world globally. It is a huge burden on our society as a whole. And we've done a really good job. As you can see from this graph, we've done a great job of moving that curve for the under age of 5. So under the age of 5, respiratory diseases killed a lot of children. And over the past 20, 30 years, through vaccinations, through public health programs that looking at hygiene and other things, have made a huge difference. You can actually see a sharp decrease in the number of cases that have occurred in children under 5. And actually in young adults and adults, it's also been a sharp decrease. But the one that has not changed much is the impact this has in the elderly, those over the age of 70. This is the major impact. And over the next few years, what we're going to see is we're going to see more and more, if we don't take care of this virus, and we, of course, hopefully nothing else emerges, we will actually see our pre-1960 levels of infection and death caused by pneumonia back in that group, age group. So this is really a big impact on society as a whole, especially with an aging population. So using the past understanding of the virus, we've been able to collect a lot of new data as well about COVID-19. We're now in a position where we can actually understand the disease, its manifestations, so being from asymptomatic to bizarre kind of clinical symptoms, and then of course the morbidity and mortality seen in the hospital situations. So we're learning more about the virus every day and some of the consequences of the virus. The one thing I tell a lot of my family members and my friends is that these are not new things. We see this with every disease. As more and more people are infected, you see more and more ranges of disease manifestations. But they are interesting, and they tell us more about how to diagnose the disease without diagnostic tests if we need to. There are actually ways to differentiate that. But it all comes down to risk. And one of the first things we teach people in the laboratory is about risk assessments and how to do personal risk assessments. And it's all about where you are and what you're doing. So we now understand the person-to-person spread of this virus, that within 6 feet of someone without a mask, you're at risk. We know it's transmitted through respiratory droplets, and that's the primary method in which it's being causing disease. And then we also know that you can actually spread the virus without feeling sick, that asymptomatic disease exists, and that you can actually spread the virus before you actually have symptoms. This is, again, not uncommon amongst viruses. It happens quite often. And then we also know that there's a potential for fomite transfer, and the virus can actually cause some transmission from one person to the next by touching something that the other person who contaminated. So these are all ways in which you can spread the virus. And now we're understanding the risk factor associated with that. So, 3 months ago, it was very difficult to know, if walking in the park was very dangerous. And now that's a lower risk than, for example, than socializing and going to a nightclub, which we're seeing over and over, infections occurring in these kind of social settings where people are inside and close together. And the good news about this allows us to understand our risk factors and allows us to make choices. So if you have a comorbidity or you're someone that may exhibit a fatal outcome or a bad outcome from disease, you can then adjust your lifestyle around that to decrease your risk. This is extremely personal, and it's behavior change that has to happen. And so, again, something we teach in the laboratory on a daily basis is learning how to do your personal risk assessments. And this is a nice slide that comes from Business Insider. I thought it was a nice way of looking at risk and where you need to be careful if you're traveling or if you're going out. And what's really interesting, if you look at the current spikes of disease, the spikes are occurring on a 7-day interval in most parts of the country. That means if you actually time that, it's on the weekends. So folks are actually probably getting exposed on the weekend and then getting disease during the week. So probably behaviors on the weekend are probably associated with this epidemiological issue that we're seeing at the moment. We'll learn more as time goes on, but there is a 7-day trend going on in most parts of the country. So transmission probability is a kind of a key thing. I bring up the mask question because this is a great graphic showing you the value of a mask. Without a mask, you have a 70% chance of getting the virus, even with a mask on. Sorry, this is actually a little bit backwards. That's sorry, my slides here are a little messed up. So if you're not wearing a mask and you're with somebody who is a carrier, you have a 70% chance of getting infected. If you have a mask and you're also wearing a mask, the risk goes down to 5%. And then if you're a carrier and you actually have a healthy person with a mask, it really reduces it down to 1.5%. So it's really a key thing is to make sure you're wearing a mask if you're sick, and make sure you're wearing a mask if you want to protect yourself against infection. On the other side of this slide really is a kind of a key graphic that these are tools that we use in the laboratory again all the time, is how do you measure your risk to disease? You measure your risk by basically 3 factors. First, your age and your comorbidity status. And then second is your exposure level. So what is your chances of being exposed? And so these are kind of key components within this process. And then the last one, of course, is the how do you protect yourself against this? So exposure, age, and health condition are in this slide set. So if you are of a high risk individual, so age and with comorbidity, and you're actually coming into contact with people who have coronavirus, you're at high risk of contracting the disease and having a fatal outcome from the disease. However, if you're working from home and you're following strict physical distancing, and you are one of those folks that's in the high risk category, your chances of infection are pretty slim at that point. Risk is always, I get asked this question, I got asked it this morning. I was training someone and they asked me, is there anything that's never no risk? In life, there's always a risk. But in reality, the risk goes down significantly to this 1 or less than 1% is what we're shooting for. So this is a nice tool to use, and we'll be sharing these slides for you to use later on. So really the first steps that we had started looking at this virus back in January, it was late December, I started watching this virus with a couple colleagues. We started watching it, wondering if this is the coronavirus that we've all been wondering would eventually emerge from bats or from some other animal. It looked very interesting. The transmission rates were pretty high, and we started to see some sequences in early January. At that point, we knew that there was actually a real chance that this coronavirus would move forward. And at that point at MRI Global, we started looking at whether or not we could develop diagnostic tests. So we started to look at the CDC assay. We have CLIA facilities where we could actually run these assays. And of course, we wanted to make sure we're ready to do that. But a lot of our commercial partners came to us and said, hey, we need to start preparing for the emergency use authorization from the Food and Drug Administration to get our assays up and going. And so we started moving in that direction, helping them do the testing that was required for actually emergency use authorization from many of the commercial partners. Later on, we obtained a BARDA proposal. This is BARDA funding actually to start looking at whether or not we could develop treatments and therapeutics and vaccines, animal models and assays. And we're now part of that international effort. It's a national, international effort to do that. That's the Biomedical Advanced Research and Development Authority, who's the key organization that's responsible for the coordination of COVID response in the US. So we're part of that, part of the WHO efforts. So we're really working towards looking at prevention methods and treatments. And as time went on, we started to actually really get focused on training, helping some of our partners who are non-laboratorians think about how they could reopen their organizations. And we've been doing a lot of that type of effort in helping understand the risks, developing pandemic response programs. And lastly, of course, using our new laboratory systems and our containment systems. The containment system we used for the Ebola patients was used for the patients that were on the cruise liners that were actually brought back to the US. So we did see utility of our devices that we're working on there. We're now working on developing some new approaches to doing this type of response, and now we're working on treatments and vaccines for government agencies, also for commercial partners. So we're seeing a lot of progress that's happening very quickly. We're doing a lot of safety and biosafety and security training as well. So a lot of efforts for our team. Our team's been very busy despite the shutdowns. So even though it's been very busy, I will tell you there's been a lot of different tests that you can have. This is a big area of concern. We'll have a whole series on, a whole session on the testing. But to give you an overview, there's molecular tests that are currently being used. Those are the most sensitive and specific assays to detect virus while you're infected. And so they're very, very sensitive, can pick up low levels of antigen or virus present in the saliva. And then of course, those are the kind of the standard tests for doing a diagnosis if you're symptomatic or you're wondering if you've been exposed or wondering if you're infected. If you want to know if you've been exposed, which means you've been exposed to the virus and you actually think you may have back several months ago, then you can actually look at your immune response to that virus through a serological assay. What that assay does is it detects the antibodies to the virus that are very specific, and those assays are now available, and there's emergency use authorizations for those. And again, these assays look at whether or not you've been exposed and you've developed immunity to the virus. Developing immunity to the virus is a very difficult question to answer. We still don't have enough experience with the virus to understand what immunity means, and so we're still working on that. And then lastly, antigen tests are coming out now, and these are like the pregnancy test of assays. They're very simple to run. You can run them in any kind of location, including a doctor's office, or in the long term, you could use them at home. But these assays allow you to detect whether or not the pathogen, the virus itself, is in your saliva by putting a drop of your saliva onto a cassette that actually gives you a positive or a negative. And these are new tests. They're usually fast, they're rapid. Typically, if you're positive, then you can be treated. If you're negative, you sometimes still need a molecular assay to tell you whether or not you're negative. So good assays and part of the repertoire of assays that we developed over time to do this. So as we move forward now, really we're now in the process of recovery. Again, as I mentioned earlier, recovery phase takes a long time. We want to make sure that folks understand that it does not happen overnight. We're now working on new diagnostic testing. So in the fall, we expect to see COVID-19 infections alongside other common cold and of course, flu infections. This will complicate the clinical diagnosis of disease because sometimes the disease is very similar in some patients. So we need assays that are considered what they call respiratory syndrome assays. These are assays that can detect all the different pathogens. And we're working with our commercial partners to develop this. The CDC just yesterday announced that they have a flu A and a flu B and a COVID-19 multiplex assay. And so we're moving towards this ability to tell you if you're symptomatic and you have a fever, whether or not you have one of these other pathogens. And these other pathogens are just as dangerous as COVID-19 sometimes for some patients. If you have comorbidities, it's important to know if you have flu A or B. It's also important to stay home. So just because you have flu A doesn't mean you can go back to work or back to your normal social situations. You should stay home for 14 days to allow that virus to disappear. It breaks the chain of infection. So again, these are really, really important assays. And ultimately, we're working on assays that could be taken at home. So we're working at home kits that could be a test that could be taken either through prescription or through a test that you could actually get through the over the counter, through the pharmacy. You could take that test home and take the test. So we're working with companies and organizations to develop those tests right now. And as we work on preventions and treatments, we're now testing those in animals and also in vitro models to assess their safety and their efficacy. We're working on assays to understand individuals who are in clinical trials of how they're responding to the virus. So we're working on those as well. That's part of this process. And building in this resilient model to how do we go back to work? How do we actually go back into the workplace and actually be resilient. When I say resilient, this new normal, how do we know if the cases go up? How do we know when to make the trigger to say, okay, everybody go back working at home, or those with high morbidity or comorbidity issues stay home, and having a kind of graduated system for allowing this to happen. And that's our next step in that area. And of course, we're working on biosurveillance and decontamination. So we're looking at ways to detect the virus in the environment, and also looking at the sequences of the virus. We're working both nationally, locally, and also globally to identify the differences in the virus and actually identify differences. The virus does change over time, but these changes are usually not significant that lead to failures of vaccines or treatments or diagnostics, but we're monitoring for that, and that's an important part of what we do in the future. Oops, slide. So as I move to this last slide, likely the most important slide that I have out of all the slides here is that you're part of the solution. And I think that's very powerful news, that we can actually be part of the solution by just changing behaviors. Washing your hands is so, so important. I can't stress that enough. You'll be surprised if you actually learn from a laboratorian how to wash your hands, or even a surgeon, even better, of learning how we don't wash our hands properly. There's some kits you can do. There's a glow germ, a product that you can actually put a lotion on that glows under black light. You can really see that you have to be really cautious about how you wash your hands. It's one of the first things we teach people how to do in the laboratory, other than risk assessments. So again, wash your hands. It's very important. Of course, the soap and waters, if it's not available, you can use the sanitizers. I'm not a big fan of sanitizers, but sometimes you have to use them. Wearing a mask, it's important. If you're not feeling well, or if you, first of all, stay home, but if you're out and about, wear a mask. It makes a huge difference in transmission. Unfortunately, it was not emphasized in the early part of this outbreak because there was a shortage of HEPA filters, and there was a conscious decision to limit the public notification of that, because they were afraid that people would actually hoard those, and they actually did hoard masks at the time. But this is an important part of our practice of protecting ourselves. Stay home when you're feeling ill. It's so important for 14 days to stay home. If you have the flu, you don't have COVID-19, or you just don't feel good, stay home. It's so important. And if you have comorbidities, be in contact with your clinician to make sure that you're being monitored remotely. There's no reason to go in unless you have called in by the physician, or your disease increases in morbidity and symptoms. Cleaning and disinfection at the work and at home is so important, and be sure to do that. I know a lot of institutions we're working with are looking at enhanced cleaning processes, and of course disinfection when they actually have a case, if a case occurs in their facility. And lastly, it's important to have an organizational plan for pandemic response, not just for COVID-19, but other things, as I said earlier, disease X one day, and of course having a family plan. It's important to not just assume that there's a plan in place. Think about how you're going to deal with connecting with family, and how you're going to take care of yourself when you're at home. So my friends often say I'm a glass half empty kind of guy. It's just my natural tendency. But despite this, I remain optimistic about our response to this virus, what we're doing. We're making great strides in it. We're learning more every day, and we hope to have a vaccine by the next year. We have therapeutics that are looking pretty useful for those that do end up in those situations, or in their hospital situation. And of course, we're living in a time in which science and engineering and technology is able to deliver this. I often remember Warren Buffett making the comment that we live in one of the best times to live in the world, and we do. And so even though it's been very stressful the last few months, and even I'm stressed about going outside on a regular basis, it feels like I'm going into the laboratory, this is a great time to be doing what we're doing. And you're seeing the dramatic impact of these technologies and science moving in our lives. And so the next year we'll see a lot happening, and I'm very positive about that happening. So I stop now. We have the question session. And of course, hopefully there's some questions for us to answer, and I look forward to any kind of comments.
All right, Gene, this is Dean again. Thank you very much. Nice job. We are gathering questions from the audience right now, and we've got several to go over. Before we get to that—
No question slide, Alexandra.
Before we get to that, I'd really recommend that you've got probably a question on that slide that you get asked most frequently. And I just ask you on behalf of the audience, pick one of those, and then we'll get into the audience questions too, if you don't mind.
The most common one I get asked is, where can I find data and news that I can trust? I shut down my kind of news gathering information. I don't pay attention to most of the news outlets at the moment. What I tend to do is watch what's coming out of the scientific literature. And of course, I pay attention to CDC and WHO. They have good comments and good guidance that are come out. They actually do a good job of having very complex data if you were willing to take that information in, but they also have simplified data that's very useful for those that don't want to hear all the scientific jargon. So that's one of the most common ones I see.
Very good. All right, we've got a couple questions here. These are good ones. All right. Okay, first one. There were recent reports that this had indications of being a blood disease, not just a respiratory disease. Can you explain and comment on that?
Yeah, so I'm not, the blood diseases, again, this is where we go into the area of speculation. What's interesting with this virus, we do not see it in the serum and blood typically. Of course, there's odd and end cases where someone's immunocompromised where the virus does escape. The virus is able to track to lymph nodes, and it's able to track to the urinary tract, and of course, it's been found in stool. So it's most commonly found in stool. It's likely doing that through some mechanisms that are not blood related, but through mechanisms which anatomically it ends up in the gastrointestinal tract, and of course, probably find some cells that can infect and actually produce virus there. But this seems to be very limited.
Okay. All right, another question, Gene. Which animal model or species is the most applicable to study?
Which animal model has been a big question. So there's been no animal model that recapitulates the human disease, and that being the pneumonia, and then of course the lethal outcome of pneumonia. So the closest model at the moment is likely to be the hamster model. The hamster model seems to be the model in which we see virus replication in the lungs. We see some damage to the lungs, and of course they do get better over time, so it's not a lethal model. So that's probably the strongest model. There are non-human primate models, but again, that's early preliminary data with that. And of course, we'd rather work with rodents than non-human primates if possible.
Yep, understood. Okay, I think we've got time for a couple more here. If air travel is necessary, what's the best type of mask to wear?
Yeah, so if you're not wearing a beard, an N95 does make sense. I just did an air travel for the first time. So I'm speaking from my experience. I saw quite interesting differences in what people were wearing. I personally wore a typical mask with a filter in it. I didn't wear an N95 because with a beard, it wouldn't make any difference. It's not effective wearing a beard in N95. But I did see some people wearing face shields. I actually wore glasses that were more like goggles, that lab goggles that are actually prescription to protect my eyes. So I did see some people wearing a face mask, which I thought was a pretty interesting idea. It does prevent potential contamination in your eyes during the process, because you could get infected that way as well. But most people are wearing a mask in the airport and in the waiting area.
All right, so here's another one. Yeah, we hear this a lot. Why is it said that wearing a mask protects others, but not necessarily the person wearing the mask? I think you had a graphic that went into that in some detail.
Yeah, so the primary reason for that is that when you have those droplets, there's some great visuals online that you can watch that show the aerosol coming out and showing how it affects when you wear a mask or not wearing a mask. The National Institutes for Standards and Technology actually just showed some nice graphics about how that works in real life. You can actually watch the videos of folks and see the droplet decrease that occurs. It really just reduces the amount of even when you cough or sneeze, how far it goes. And so that really, it captures it on that mask. And then if you throw it away and put a new one on a regular basis, it really does reduce the risk.
Yeah, all right, very good. All right, well, I think those are the questions that we have from the audience right now.
Great.
And we're going to move into another part of the session. And we're going to break out into discussion groups now. And everybody online, you should be able to find this join discussion group button that's below this video. And that'll open up a different Zoom application. And what we'd like you to do is, if you want to join a discussion group, be able to talk with Gene, or you're going to talk with one of our other experts, Joe Bogan, click on that button and then give us a moment. And we're going to get everyone broken out into these rooms. And then you'll be joined by the MRI Global facilitators to help continue the discussion.
Great.
All right.